Welcome to Cell and Molecular Biology. Today, we are diving into a fascinating topic, the transcription unit. If you have ever wondered how a cell knows exactly which part of its DNA to read to build the proteins that keep you alive, you are in the right place. We will build our understanding from the ground up, so do not worry if this is completely new to you. Let us get started. To begin our journey, let us think about the DNA inside a cell. Imagine you are standing in a giant library that contains the blueprints for building and maintaining an entire city. This library represents the DNA. If you only need to build a single bridge, you would not photocopy every single book in the library. That would be a massive waste of time and resources. Instead, you would find the specific book, locate the exact chapter for the bridge, and copy just those pages. Building on our library analogy, in a cell, a transcription unit is exactly like that specific chapter or recipe. It is a defined segment of DNA that contains the instructions for making one specific piece of RNA. This RNA will eventually be used to build a protein. By having these distinct, organized units, the cell can efficiently read and copy only the instructions it needs at any given moment, rather than copying the entire genome. This selective reading is what allows different cells in your body to perform completely different functions, despite having the exact same DNA. Now that we understand the concept intuitively, let us look at the formal definition. In molecular biology, a transcription unit is defined as the sequence of DNA that is transcribed into a single molecule of RNA, along with the adjacent sequences required for its transcription. It is not just the coding part. It includes the control regions that tell the cellular machinery where to start and where to stop. This ensures the entire process is highly regulated and precise. This brings us to the anatomy of the transcription unit itself. It consists of three critical regions. First is the promoter, which acts as the starting line where the enzyme RNA polymerase binds to initiate the process, though it is not itself copied into RNA. Next is the structural gene, which is the actual coding region that gets copied into the RNA molecule. Finally, we have the terminator. This is the sequence that signals the RNA polymerase to stop copying and detach from the DNA. Together, these three components ensure transcription begins and ends at the precise locations required. Let us visualize what we just discussed with this schematic diagram. You are looking at a representation of the DNA strand. On the left, in green, is the promoter region, where the machinery binds. Notice the arrow labeled +1. This is the exact starting point of a transcription. Everything to the left of this point is called upstream, and everything to the right is downstream. The blue section is the structural gene, the actual code being copied. Finally, the red box on the right is the terminator, where the process stops. This beautiful organization keeps the cell functioning perfectly. Having seen the structure, you might wonder why understanding this is so important. The transcription unit is the fundamental basis of gene expression and regulation in all living organisms. Without these clearly defined units, RNA polymerase would randomly copy DNA, creating useless fragments and wasting massive amounts of cellular energy. In real-world medicine, many genetic diseases and cancers are caused by mutations within the promoter or terminator regions, leading to abnormal protein levels. Furthermore, biotechnology relies on this concept to insert artificial transcription units into bacteria, allowing us to produce life-saving drugs like human insulin. Before we calculate the size of the RNA produced, we must understand how biologists map the DNA. They use a specific numbering system based on nucleotide positions. The exact nucleotide where transcription begins is designated as +1. Any position before this start site, moving backward along the DNA, is considered upstream and is given a negative number. Conversely, any position after the start site, moving forward through the gene, is considered downstream and is given a positive number. There is no zero in this system. It goes directly from -1 to +1. This numbering system leads us directly to our first mathematical representation, calculating the length of the RNA transcript. The formula is lRNA equals n term minus n start plus 1. Conceptually, we are just finding the distance between the start and end points. lRNA represents the total length of the newly synthesized RNA transcript, measured in nucleotides. N term is the exact nucleotide position where transcription terminates. N start is the position where transcription begins, almost always +1. We add 1 at the end to account for inclusive counting. If the termination position increases, meaning it is further downstream, the total length of the transcript will proportionally increase. Let us see this formula in action with a concrete example. A researcher is studying a bacterial transcription unit. We are given that the transcription start site is at position +1, and the polymerase releases the transcript at position +1250. We need to calculate the total length. We choose our length formula and substitute n term with 1250 and n start with 1. Subtracting 1 gives us the raw difference, and adding 1 corrects for the inclusive boundary. The final answer is 1250 nucleotides. This means the structural gene successfully coded for a molecule exactly that many building blocks long. Now that we have calculated the length of the transcript, we can determine how long the transcription process takes. This comes from the basic physics principle that time equals distance divided by speed. Our formula is T equals lRNA divided by R poly. Here, T represents the total time required to transcribe the gene, usually measured in seconds. lRNA is the length of the RNA transcript in nucleotides that we just learned how to calculate. R poly represents the rate or speed of the RNA polymerase enzyme, measured in nucleotides per second. If the gene length increases, the time required increases. Conversely, if the polymerase rate increases, the time will decrease because the enzyme is moving faster. Let us apply this time formula using the RNA transcript from our previous example. We know the transcript is 1,250 nucleotides long. We are asked to find the time required for transcription, given that the bacterial RNA polymerase travels at a constant rate of 50 nucleotides per second. We use our time formula, dividing the total length by the rate. We substitute lRNA with 1,250 and R poly with 50. Dividing these gives us 25. The units of nucleotides cancel out, leaving us with a final answer of 25 seconds. Physically, this shows how incredibly fast cellular machinery operates, allowing bacteria to respond to their environment in less than half a minute. We have covered a lot of ground today, so let us review our key takeaways. First, a transcription unit is the fundamental functional segment of DNA that gets copied into RNA, acting like a specific recipe from a massive library. Second, it relies on 3 main components: a promoter to start, a structural gene for the code, and a terminator to signal the end. Finally, we learned how to mathematically model this process, calculating both the physical length of the transcript and the time required to synthesize it. You now have a solid understanding of how cells precisely control and execute gene expression. Great job!